Hey everybody, welcome. We're going to look at chapter 9 today. Chapter 9 is all about the cell cycle and how cells reproduce or how we get more cells. So if we're thinking about sort of the journey we've been on in Bio 101, we first looked at what makes living things, then we looked at populations and the environment, and then we sort of shifted back and we took a look at a little bit of chemistry, inorganic, then organic, then parts and pieces that make up cells, then how cell parts work, so how the membrane works and how cells get energy. And now we're really looking at how cells make more of themselves. And this chapter is not as intense or in depth as the photosynthesis and cell respiration stuff. I also think students have seen this stuff before, even if it is something you don't really remember. That being said, there are going to be lots of vocab words between the lecture and lab, so I highly recommend the study guide and the Quizlets as some good ways to study and to see things um, that you'll need. So let's go ahead and jump into it. We're going to start with the cell cycle. What is it? What does it have in it? Then we're going to look at eukaryotic chromosomes and DNA and how that's compiled. Then we're going to take a look at mitosis all the way through to cytokinesis. So mitosis being the nuclear division and cytokinesis being the cytoplasmic division. Then we're going to look at the cell cycle and what it means in cancer. And then very briefly talk about prokaryotes and how their cells divide. So let's take a look at the cell cycle. The cell cycle is this set of orderly stages that a cell is going to go through from division, then through time, living its cell life, and then resulting in two new daughter cells. So two new cells. It is going to include just prior to the next division that it happens, where the cell gets larger, we double the number of organelles, we make a copy of the DNA, and then it's going to have interphase, which has several parts in it, and then the mitotic phase or mitotic stage, which has mitosis, which is broken into parts, and cytokinesis. So the cell cycle as a whole is just the cycle of growth and development of a cell until it divides. We follow it through, and then what happens once that cell has divided and it grows and does its cell stuff that it does. If we look at it, we can look at it in two chunks of interphase and then mitosis with cytokinesis. Interphase is just this normal living its cell life. There's going to be a growth phase, we call that G1. Then there's going to be an S phase where there's synthesis and DNA replication. So we're copying our organelles, we're copying our DNA. Then there'll be a growth 2 phase where we do all the final things in preparation for the cell to divide. Then we'll go through the mitotic phase, which includes prophase, prometaphase, metaphase, anaphase, telophase. And then we go through cytokinesis, where we're splitting up the cytoplasm and the organelles. And then it heads back into growth. What you'll see is that in this, there are certain checkpoints or stop points. There is this checkpoint here after growth, where basically if something is wrong with the DNA, this cell will go through apoptosis or be destroyed. After growth 2, same thing. There's this checkpoint or stop point where before mitosis occurs, we're double checking, is the DNA correct? If not, then that cell will go through apoptosis. And then there's this mitosis checkpoint or M check or stop point when the spindle fibers are all assembled and the chromosomes are lined up before they are pulled apart. If they're not lined up properly, we will stop mitosis. So let's take a look then at these chunks of time the cell is in, interphase or mitosis and cytokinesis, and see what's happening. So we can have interphase. Interphase is where most of the cell's time is spent. This is when it's doing whatever its normal cell job is. The amount of time that a cell is in interphase is going to vary on the type of cell we're looking at. For much of what we're going to do, we're going to look at just the generalized cell, but we know from learning our cell organelles that some cells have more of a certain type of organelle or less of a certain type of organelle or don't divide, things like our nerve cells. So know that some of what we say is that generalized version, and it will depend on the cell type that you are looking at. A good example, nerve cells and muscle cells don't complete the full cell cycle. So they are going to be in sort of a different part of the cell cycle that we call the G0 phase. They're not going to go through the growth and synthesis and growth, then mitosis, like other cells do, like a skin cell or a stomach cell. So that phase is called G0. And then there is the G1S and G2 phase. This whole part of interphase, G1, S, and G2, is about 90% of the cell cycle. So when you look at cells under a microscope, most of them, most of the time, are going to be in interphase. Most of them, most of the time, are going to be in interphase. 90% or so. What is happening during these phases? In G1, the cell is sort of in this recovery mode from its last division. Think of a track star that just ran a race. They don't try to run another race right away. They'll take a break and get water and stretch and maybe wait a day or two or sometimes weeks before they run, another marathon. That's the recovery. Also in G1, cells are doubling their organelles. So they're making copies of their organelles. And the cell is getting bigger. The cell is also building the building blocks or accumulating the materials that it needs to make more DNA or synthesize a copy of its DNA. Then we'll switch over to the S phase. That's the middle part of interphase. In that S phase, DNA replication is happening. We need to make proteins that are going to help with DNA synthesis. Our chromosomes will enter this S phase with one chromatid, so they will condense from a bowl of spaghetti looking stuff into like a little gummy worm. And then that chromatid is going to be copied. So we're going to leave S phase with two identical chromatids. So they are going to then be called sister chromatids. And then sister chromatids are going to remain attached until they separate during mitosis. So if you've looked at chromosomes before, you probably saw like a little X shape. Those little X's individually, that is a chromatid. Together in that X shape, those are sister chromatids. Then during G2 phase, we are going to have this sort of shortened phase between DNA being replicated and mitosis starting. But that phase is when We're going to make any more proteins that we need. We're going to make our microtubules that we need. Anything that we just need some more of is going to be made then. And then we'll go through the mitotic stage or M stage. Mitosis refers to the nuclear, the nucleus division. Cytokinesis is the cytoplasm and organelles. So when we say mitosis, although we will describe the whole cell in much of this, we're really just talking about where is the nucleus, where are the chromosomes. In mitosis, those daughter chromatids or sister chromatids are going to be distributed and split up and become two brand new nuclei in two new cells. And cytokinesis, of course, is the division of the cytoplasm. When we go through mitosis, we are getting two genetically identical daughter cells. We are making an exact copy. Think of it like a Xerox machine, a copier. You put your paper on it, you push the print button, and it makes an exact copy of the original. That is mitosis. We will see that there is another M word for division, meiosis. They sound similar, but that is for sexual reproduction, sex cells, and they will not be the same. But in mitosis, they are the same, identical. With all of that, we want to remember that there is this controls in the cell cycle because things could happen along the way that we then don't want to pass on to new cells. So there is a signal, and that is just any molecule that will influence activities. But we can have a signal to stop cell division. And that signal can be controlled by internal and external factors. External are outside things. So there's growth factors that are signaling proteins received at the plasma membrane. So remember our plasma membrane, it is going to have those receptor proteins to get molecules, one of them being growth hormones or growth factors. If they don't get those, the cell's not going to continue in the cell cycle. Then some internal things, there are proteins called cyclins that will increase and decrease throughout the cell cycle. And you have to have certain cyclins at certain points for it to go on to the next stage. If we don't have cyclins, we stop at G1, G2, or mitosis. So those are those little checkpoint stop signs. Some examples, if there is solar radiation, like from the sun or from X-rays, then that could mess up our cyclins for that G2 checkpoint, and then the cell cycle does not continue with that cell. So it's a protection that we are making that identical copy. And we want that identical copy. If we're making a skin cell and we want to make another one, we want it to be exactly perfect. We don't want it to have any errors that could be passed on. And so we won't copy cells that have errors and are paused. How do we get rid of those paused, error-filled cells? Something called apoptosis, which is this programmed cell death. It is this very orderly way in which a cell basically self-destructs. First, the nucleus will fragment apart. Without that control center, it's only a matter of time until the rest of the cell falls apart too. We'll see blistering or like this little bubbling of the plasma membrane. Without a plasma membrane controlling what is coming in and out of the cell, again, it's just a matter of time before the whole thing breaks apart. We can engulf these parts and pieces of the cell in white blood cells or other cells. Those lysosomes and peroxisomes of other cells can break apart things, and then the cell is destroyed. Apoptosis primarily is caused through enzymes called caspases. They're the things that are breaking apart, fragmenting the nucleus, blistering the plasma membrane, destroying the parts of the cell. And really, mitosis and apoptosis are these battling, competing, opposing forces. Mitosis increases cell numbers, apoptosis decreases cell numbers. So we can see here, for example, these are normal cells, skin cells. They want to make a copy. They start making a copy and something went wrong. So we've stopped at our checkpoint. And now we'll see that this once sort of fuzzy cell is going to bubble up, puff up. The nucleus is going to start to fragment and collapse. And then there's these little blebs from it bubbling. And then it can be gobbled up by white blood cells, broken down by cell parts. And we have these little fragments that destroy this cell. Cell division and apoptosis are always happening throughout the lifetime of an organism in order to maintain the body's homeostasis, right? We need certain cells to live. We need some cells that are old or worn out or are incorrect to die, and these processes do that. An abnormal cell could become cancerous if it becomes infected to a point where it doesn't die through apoptosis. It could be from a virus. It could be from a mutation in the DNA. It could be many things that make it not go through apoptosis. Once it doesn't go through apoptosis, it'll just keep dividing and dividing and dividing. And that's what we know of as cancer, as a tumor. Cells can have capsase enzymes within them all the time, but they're normally kept at a balance. There's inhibitors that check them until they're needed. And so then they're unleashed when that cell needs to go through apoptosis. There is a signaling protein called P53. That is what is going to stop the cell cycle at G1, and that is when the cell's DNA has been damaged. If we can repair that damage, that is what will happen. And if that repair is successful, we'll keep going through to go to mitosis. But if not, then our apoptosis will be initiated. So here's how that's working. These proteins and enzymes aren't super, super important. Like, don't spend all your time memorizing them. But for those of you that really want to know how this is all working, this is great. So you will see in our top picture here. In our top picture here, we have a kinase protein, an enzyme here. If it's not present, then we have this factor that's connected here, and we can stop everything from happening. But then we can add phosphorus with this special other protein. This cyclin-dependent kinase enzyme adds phosphorus, releases our factor. Our factor can then go and bind to our DNA. Down here, if we have our P53 protein, DNA is damaged, then we're going to get a phosphorus. We're going to try to repair this DNA. And if we can repair it, great, then we'll continue on in our cell cycle. If we can't repair it, we're going to go to apoptosis. If we read our DNA and there's no damage, then we'll just break down this P53 into its components. We can build it back up when our cell is going to divide after it has lived its cell life. So when we look at chromosomes, which we'll do shortly, we know that that is going to be made of DNA, which is our nucleic acid and our double helix. We know from learning about organelles that is in the nucleus as chromatin. Chromatin just sort of looks like a bowl of spaghetti of DNA. We will start to wrap our DNA, our chromatin, around these little proteins called histone proteins. When DNA wraps around it, then we call it a nucleosome. When we start to organize, bend, and fold those histones with DNA around them, we can make what we call euchromatin, and we can keep compacting until we get our chromosome that we have seen and know as these little sort of worm-like structures. One arm of this is a chromatid, and this is the sister chromatid. When they break apart during mitosis, they're still going to be our chromatids, just apart, and they are also chromosomes. It's kind of confusing, the language, so there is a video for you demystifying chromosome number that might be very helpful if this process is sort of confusing, but know that this is one set of your DNA. We made a copy of it over here. They're matching sets. So if this chromosome coded for eye color, this other side is also coding for eye color. It's an exact copy. And those are your sister chromatids that make up your chromosomes. So in word form, of course, your chromosome is going to be this long singular DNA molecule, which we know is a double helix. It is wrapped around histones, which are a type of protein. And when we have our DNA and our histone proteins, we call it chromatid. Histones really are just this structural component. There's 5 different kinds mainly, but really the thing you need to know is that they're going to pack the DNA into the nucleus neatly. DNA then just gets wrapped around 8 histones to make a nucleosome, which kind of looks like a little bead. Then nucleosomes are linked together by portions of our DNA. So it sort of looks like beads on a string. Then that folds in a zigzag compact structure, and then it loops and is coiled. And then euchromatin is this part that has been used by RNA polymerase to make proteins out of. But essentially, we have combined it, wrapped it down, and made it very dense, as opposed to like a bowl of spaghetti noodles that have been cooked. This is nice, organized spaghetti noodles in the box. The more highly compacted form of chromosome is called heterochromatin. This is not used by ribosome, so it's inactive. These genes don't usually get transcribed to do anything. And it's just more easy to move your chromosomes when they're compacted than when they're loose. And most chromosomes have both this looser compaction where ribosomes can transcribe and this tightly compacted that is not used DNA. So there's a mix on all of your chromosomes. All right. The division is dealing with this nucleus and these chromosomes. Where are they going? What are they doing? And then cytokinesis is the cytoplasm. Where is it going? What is it doing? As a whole, during mitosis, these sister chromatids are going to line up in the middle of the cell, then be separated, and distribute it into what will become 2 new daughter cells. So before mitosis can even begin, chromosomes have to be formed. So we have to condense chromatin into coils to make chromosomes. And it is important to note that each species has its own number of chromosomes. The number does not make you more intelligent or less intelligent as a species. You just have an amount of chromosomes. For example, fungi like yeast have 32. Garden pea has 14. In animals, fruit flies have 8. People have 46. Goldfish have 94. So it is a number of chromosomes, but every time your cell divides, your cell has to compact your DNA into these chromosomes. When we compact this DNA, we can count the number of chromosomes, and we can see if there is a diploid or a haploid number. Diploid is also written as 2N. Diploid is when there are 2 sets of chromosomes for each chromosome that you have. So humans, we have 23 different types of chromosomes. Each type is represented twice in body cells. It's diploid. Or you can think of it as getting one from your mom and one from your dad for all of your chromosomes. So total, you have 46 chromosomes. One copy of all of your genetics comes from your mom. One copy of all of your genetics comes from your dad. That is a diploid cell. So when we're looking at body cells, we will see those 46 chromosomes. That is going to be diploid. Haploid is when you have half of the number of diploid. Eggs and sperm have half the number of chromosomes. They only have 23 chromosomes. So that is haploid. When we look at the math of that, 2N is 46 in each nucleus, one set from the dad, one set from the mom. If we look at sperm and eggs, we would just see 23 chromosomes present. During interphase, the cell is preparing to do mitosis. Organelles are duplicated, and by the end of that S phase, synthesis phase, each chromosome has been duplicated. Then we will have 2 identical double helix DNA molecules for each type of chromosome that you have. Then we connect those matching kinds, sister chromatids, and we're going to attach those sister chromatids together at a central point called the centromere. During mitosis, centromeres holding the sister chromatids together will separate. The sister chromatids separate, and then each of those sister chromatids will then be called a daughter chromosome. Daughter chromosomes will then be distributed into 2 new daughter cells and make up their daughter nuclei. So again, here's our sister chromatids. This middle part where they're together is called the centromere. It will have this accessory protein here called a kinetochore. That's where we're going to attach these spindles, these little fibers that will pull this apart. So just outside of the nucleus is something called a centrosome. Centrosomes is this microtubule organizing center. this little control box for microtubules in animal cells. We made a copy of this during the S phase, so now there are two. We can think of these sort of like this barrel-shaped thing in animal cells, and they're oriented at right angles to each other. And we call them centrioles. What's going to happen is those are going to migrate away from one another, and they contain those microtubule fibers or spindle fibers. Each fiber is going to then, again, be made of microtubules, which is going to be made of tubulin, and they are going to change the shape of the cell so that it can be ready to divide. What does this all look like? Well, there's phases or step-by-step of how this is happening in mitosis. So we go from interphase, where we have growth, synthesis, growth. Now we're in mitosis. We're going to start with prophase. Prophase is when this nuclear division is just about to occur. Chromatin condenses. You can actually see the little X shapes when you look at the microscope. Each chromosome is held together at its centromere. The nucleolus is going to disappear. The nuclear envelope is going to disappear. We'll start to see the spindles, little strings assembling. And those centrosomes, the centrioles, they move apart from each other, going to opposite ends of the cell. In animal cells, when that starts to happen, it makes this little star array, so it's sometimes called an aster. Then we have this sort of in-between phase called prometaphase. It's like a mix of prophase and a mix of metaphase. And this is where we're really starting to organize those sister chromatids because they're going to be separated soon. The centromere in each chromosome is going to have its kinetochores, which is these proteins, and we're physically connected still with our sister chromatids. At those kinetochores, we're attaching our spindle fibers. Those spindle fibers are then again connected to those centromeres, the centrioles that have spread apart to opposite ends of the cell. In metaphase, I like to think M for middle, M for metaphase. The chromosomes are being pulled by those fibers, and they are aligned in the middle of the cell. We call it an equatorial plane, or think like the equator is in the middle of the earth. This is the chromosomes in the middle of the cell. Sometimes you'll hear this called the metaphase plate. That just represents this 3D plane where the cell is going to be divided. There is a checkpoint here that will pause everything until all the kinetochores are attached and everything is lined up and organized. Then anaphase will happen. That's when the centromeres dissolve. It releases the sister chromatids. So now they are separated and they are pulling away. A for anaphase, A for away. Now, what used to be called our sister chromatids are each called daughter chromosomes, and they are being pulled in opposite directions by those microtubules, or what you had called kinetochore fibers. The stringy things that are attached to the chromosomes are pulling them, and the poles are moving farther apart from one another. Then you have telophase. Telophase is when those spindles will disappear because our chromosomes have sort of reached the opposite edge of the cell. We're going to start to get a new nuclear envelope forming around those chromosomes. And now there are two definite clusters of chromosomes. You will still see that there are two of each type of chromosomes, and this cluster is where this nucleus is going to form. And then the division of the cytoplasm has already started during telophase, but we know that that's called cytokinesis. Once we have that new nuclear envelope, those chromosomes will start to relax and become chromatin again. So what does this look like? So we have our cell. It is right before this in interphase at that last growth. Then we are going to have our chromosomes condensing. We are going to have our centrosomes moving around in the cell, starting to go to the poles. The nuclear envelope is going to disappear. Our centrosomes are going to connect to these kinetochores on these condensed chromosomes in prophase. In prometaphase, we're starting to line them all up, and that connection is being made. And then in metaphase, they are all lined up in the middle. Those chromosomes are in one straight line. There's a checkpoint here to make sure everything is lined up and that those kinetochores are attached to those fibers. And then in anaphase, they pull away. A for anaphase, A for away. And then in telophase, we now have those chromosomes in the two new daughter cells. So they've pulled all the way apart. We've started to reform a nucleus around them, and they will start to relax. This process is the same in plant cells, just that there is a cell wall that you will see very stationary while this dividing, pulling apart is happening. And then we'll see once we get to telophase, there's a little bit of a difference. So again, here is this early prophase. We have copies of our centrosomes, of all our organelles, truly. And the chromosomes are starting to form from chromatin. In prophase, we see that the nucleolus has disappeared. We're seeing that there are very, very visible chromosomes. In prometaphase, we have that kinetochore is attaching to those spindle fibers, and now we've got our centrosomes at the poles. In metaphase, now the chromosomes are all lined up in the middle of the cell. They're all attached to spindle fibers. In anaphase, the sister chromatids are pulled apart and become chromosomes again. And then in telophase, the daughter cells are forming new nuclear envelopes, nucleoluses, and the chromosomes will start to relax into chromatin. The last part of this is cytokinesis. That is the division of the cytoplasm. That is going to allow that starting cell cytoplasm, that mother cell, to equally distribute its cytoplasm to the daughter cells. It is going to happen when this plasma membrane pinches in or encloses on each daughter cell, and now we're left with two new cells. Truly, this starts in anaphase. It's very present by telophase, but it is its own process, right? Cytokinesis is the cytoplasm. Mitosis is the nucleus. It is going to be slightly different in plants and animals. In animals, we'll make a cleavage furrow. It's this little pinched in area between the daughter nuclei or the two new nuclei that you make. There'll be this contractile ring made of actin, and it pulls like a little drawstring, and it pinches the mother cell into two. can almost think of it like putting a rubber band around a water balloon, around the middle of it, until now you've made like 2 new water balloons. In plant cells, we can't just pinch in like a water balloon because we have our cell wall. So we're going to make a cell plate that is going to be made from the Golgi body or Golgi apparatus, sending out vesicles that will line up in sort of a midpoint of the cell. And then eventually, all those vesicles, it'll fuse into this thin membrane and then a cell plate. Or I guess those fused Golgi vesicles become that cell plate that then will become the plasma membrane separating the two cells. That little space in between is called the middle lamella, and then there will be cells that will make cell walls on the outside into that middle lamella, which will then become the cell wall. So here's it in animal cells. We're kind of pinching in, pinch, 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 like wrapping a rubber band around and around and around, or pulling a drawstring and cinching it to make 2 cells. And here's what we're looking at in plants. We're making these vesicles. They're going to fuse and make that cell plate, which will then secrete, or cells there will secrete to form the cell wall. Okay, what is the purpose of doing this in cells? Well, we need mitosis for growth and repair. So, like if you cut your skin, you need to replace that cut skin with new skin cells. Also, if you want to grow taller or wider, you will need your cells to divide. In flowering plants, there's a special type of tissue called meristematic tissue that is able to divide the entire lifetime of the plant. So many plants, they have what is called indeterminate growth, meaning they'll grow as big and as wide as time allows them to. Trees, for example, have to increase their width in each growing season. In mammals, mitosis is necessary when an egg becomes an embryo, so it's been fertilized, when that embryo becomes a fetus. After birth, the child will grow and become an adult, and also to cut or to heal cuts or broken bones. Stem cells are going to be, in many mammals, they are the part of the plant or a part of the animal that is retaining this ability to divide, just like our meristem tissue in plants. For example, red bone marrow stem cells produce all kinds of types of blood cells. So there was a push in the 1990s to do a lot of stem cell research that has, in more recent times, gotten a lot of backlash. But the technologies to grow organs and to make blood without blood donations is something stem cells are working towards. So therapeutic cloning can be to make tissues that can begin with adult stem cells or with embryonic stem cells. We can grow organs not on a human. And then embryonic stem cells, that's where much more of this controversy comes from, used for reproductive cloning. So to make a person. So that's like the in vitro fertilization methods use embryonic stem cells. The controversy of these come in, one, are we sort of playing God by making, a liver in a lab or an ear grown on a mouse that we can put on a person? And two, what happens to the stem cells that we do not use, specifically the embryonic ones, when you do something like in vitro fertilization, you don't put all of those embryos into the person to hope they attach to the uterus. So what happens to them um, is an ethical issue for sure. You may have heard of Dolly the sheep, and how that worked was that there were stem cells. Um, and so we can take some G0 type cells, things like muscle cells, brain cells. We can take the nucleus out of those cells, fuse it with the nucleus of an egg, put it in the egg, and then now that's going to develop cells that you can then implant into a surrogate mother, and then a clone is born. So with Dolly the sheep, they took Dolly's cells, removed the nucleus, put it into an egg that then grew and became Dolly the sheep that was born as a clone. The other way that you can do this is you can take the nucleus, fuse it into an egg, then you can get these embryonic stem cells, and then you can stimulate it to become something, become nervous tissue, become blood tissue, become muscle tissue. And then you're not making an entire um, organism, you're just making the parts and pieces that you want. Um, so pretty cool. There is a lot of research being done on this still, though the push and funding isn't as great as it once was. Um, it's still being used all the time. And I encourage you to do more research if it's something that interests you. All right, so we said that the cell cycle has this sort of checks and balances stopping points, that it won't let cells that are damaged in some way divide. It will go through apoptosis. Then we said with apoptosis, there are cells that can be damaged or infected or somehow able to override apoptosis. And really, what that ends up meaning is that abnormal growth in cells is what we call a tumor. And in order for those cells to grow, they go through the cell cycle. So, at its very root, very core, cancer is just this error, this mess up in the cell cycle. With tumors, you can have benign tumors that are not cancerous. They're encapsulated, they don't invade or spread. They're really just all in one enclosed. They'll get so large in that enclosed space, but they're not trying to spread out. Then there is malignant tumors that are cancerous. They are not encapsulated. They will get into neighboring tissues. They can also detach and lodge in other places. That's metastasized when it moves somewhere else and infects that place. And this is usually a result from mutations in the genes that regulate the cell cycle. The development of cancer tends to be gradual and multi-step. And there are cells in your body all the time that are, cancerous, but then your body detects that and it goes through apoptosis or is destroyed. So it can be many years before a cell is obviously cancerous. That being said, we probably all know someone who their cancer was not gradual and multi step, it was very rapid. And that can be for many different factors, um, but ultimately is this error in the cell cycle. Some characteristics of cancer cells is that they do not specialize or they lack differentiation. Um, and they are sort of these immortal cells that can enter the cell cycle repeatedly. In your body, normally, even when we clone something like that therapies, um, those cells are going to become certain things, right? We can hijack them to become nervous tissue or muscle tissue or blood tissue or grow skin tissue. Um, or it's going to become the whole organism if that's the type of um, path we chose, right? We made a whole lamb, Dolly the sheep. But if the cells don't specialize and become a nerve cell, a cell in your intestine, a brain cell, and they're able to constantly go through the cell cycle, that means they lack differentiation. And that is a characteristic of cancer cells. They have an abnormal nuclei. It could be really big. It could have a weird number of chromosomes. Often it will have extra copies of genes. It just isn't what the normal cells look like. And they don't go through apoptosis. Normally, cells that have damaged DNA, when they're going through the cell cycle, will go through apoptosis. The immune system can recognize these abnormal cells and trigger it, or those checkpoints trigger it. But cancer cells don't go through apoptosis, and so they will continue to make more of themselves. In order to form tumors, they'll have to grow on top of each other. Cells don't really like that. So mitosis is normally controlled by contact with neighboring cells or something called contact inhibition. If a cell touches another cell, it's like, cool, I can stop now. I've reached the point I need to get to. But cancer cells don't have that and they'll grow on top of each other. They can also go through that metastasizing process where the original tumor is easy to fracture and break apart, and those little parts will move around the body and can appear in other organs. And then something called angiogenesis means that they'll make blood vessels to bring nutrients and oxygen to the cells of that tumor. And these cells having these characteristics is what makes cancer so difficult to treat, because you have to remove all of those infected cells. That's why they do a biopsy and they check for the margins, so that if they have to take more of it away, because any one cancerous cell left behind will do all of these things and cancer will still be there. So normally the cell will just stop and it won't make more. But somehow this cell got hit with a dose of UV or X-ray. This cell had something happen to introduce a mutation. Then that cell is going to not be able to stop going through the cell cycle. It'll make copies and copies and copies and grow on top of each other. If it reaches into blood vessels and veins or the lymphatic system, then it is able to travel all over the body and can show up in new places. And then new tumors are found far distance from those primary tumors. So when we compare them, this is a nice table. Cancer cells are non-differentiated. They don't have special jobs. Normal cells do. We'll have weird nuclei in cancer cells, normal nucleus for normal cells. Cancer cells do not do apoptosis, normal cells will. Cancer cells do not have contact inhibition, but normal cells do. Cancer cells are really disorganized, multiple layers, where normal cells are a very organized layer. And then cancer cells can metastasize, where normal cells will just stay in their original tissue. Of course, normal growth and tissue maintenance is going to depend on a balance between signals that say, yes, cells you need to divide, or no, inhibit the cell division. And there are two types of genes that can be mutated And then this balance can be upset, which can cause cancer. This is not the only ways. Cancer is complicated, and one kind of cancer in one person may look very different than even the same type of cancer in a different person, or different kind of cancer in people. It's hard to treat, and we have not been 100% successful in wiping out cancer because of these difficulties. But if we look at these two genes, or two kinds of genes, you can have oncogenes, and there are something called proto-oncogenes, which make proteins that promote the cell cycle. If that proto-oncogene is mutated, it can become an oncogene, and that can cause cancer. There's also tumor suppressor genes, which inhibit the cell cycle and promote apoptosis. If the tumor suppressor gene is inactive, it can promote cancer growth. Both of these types of genes are normally regulated within this organism's growth plan. Like, your body and your DNA already normally has this under control, but there could be mutations introduced many ways that could turn these on or off in bad ways. If you've ever taken a 23andMe test or the BRCA breast cancer test, they have you spit in a little tube, and then send that tube off, and they can look at your DNA. And when they give you your health results, one thing they're looking at is these types of genes, and the BRCA is looking at these genes to see if you have errors that have been passed down. But just having these genes be mutated does not necessarily determine that you will get cancer either. Even if your proto-oncogenes become oncogenes, or your tumor suppressor genes become inactive, that doesn't necessarily mean you get cancer. It is just you are more likely, because everything's a little bit of nature and nurture, so it depends on your lifestyle and other components of your health as well. So if we look specifically at the tumor suppressive genes becoming inactive, again, tumor suppressor ones are the ones that inhibit the cell cycle. Some promote apoptosis, but if there is a mutation in this, it will accelerate or speed up the cell cycle. So we can have RB and P53, which we said before were part of our stop sign in mitosis. They code for proteins with these same names. Retinoblastoma is inherited because of a mutation in the RB gene. P53 turns on the expression of other cell cycle factors. And about half of the human cancers we know have a weird P53. But again, just having these does not necessarily guarantee cancer. There are other things that can influence it as well, but it does make you more at risk. If we look at the proto-oncogenes, they promote the cell to go through the cell cycle. They have receptor and signaling molecules. If they become oncogenes, it can have this abnormal protein product or high levels of the protein, and then unregulated cell division happens. But we know of about 100 oncogenes right now. The research is still out there. There could be more than that. But again, the BRCA is a mutation that causes breast and ovarian cancer. So here are some things that could happen. It could be heredity, where you're passed on these genes. I've personally taken the BRCA, as my mom passed away from breast cancer pretty young. She was in her 40s. And so I had to take the test. You spit in the tube, send it off. I was negative for these genes, but that doesn't mean that I'm not at risk of breast cancer, because radiation affects them, um, viruses can affect them, pesticides, herbicides. There's many things that can mess with these genes or other parts of your DNA and can, in fact, lead to cancer. Some other causes: your DNA, all your chromosomes, when they condense, they have these little tails on them called telomeres. And it's not that it's junk DNA, but it's DNA that's not responsible for coding any usable proteins. And so every time your cells divide, these telomeres get shorter and shorter. When they get very short, the cell will no longer divide. But as they are getting shorter, when the cell divides, it can um, mess up and not get all of the DNA that you need. So you can have mutations. You can also have mutations in the gene that codes for these telomeres, these little lengths. Um, it can cause the telomeres to lengthen, and then those cells could just continuously divide. All right, last but not least is something entirely different: prokaryotic cell division. This is for bacteria and archaea. They have one singular circle DNA, and it's all folded up into that nucleoid region. What's going to happen is we will make two rings of their DNA before the cell divides, and then that ring will attach to a point on the plasma membrane. Then the division part is called binary fission, and this is when we split the cell into two. Those two replicate chromosomes are distributed, one into each cell, and it is a type of asexual reproduction. We're making two exact copies. For example, E. coli, which is found in mammal intestines, it has a generation time where it will divide about every 20 minutes. When we compare prokaryotes and eukaryotes, we need both binary fission and mitosis to make sure that the daughter cells are a genetic identical copy to the parent. If we look at prokaryotes, some protists like algae and protozoans, and then fungus that are single-celled, cell division in a single-celled organism makes two new individuals. So we say that that's asexual reproduction. In multicellular fungus, plants, and animals, cell division doesn't make a whole new creature, but it is important for growth and repair and renewal. When we look at binary fission, the chromosome duplicates and each daughter receives one copy. And then that parent cell will just elongate and then it'll split. There's no spindle fibers. It's just this pulling apart that happens. So we'll have our bacteria cell making a copy of its chromosome. It now has two copies attached to that membrane. The cell will elongate, and then the DNA is moving into what will be the two new cells. We pinch it in together, and we end with two new identical daughter cells. So again, for prokaryotes, bacteria, and archaea, this binary fission is what is responsible for asexual reproduction. In protists and some yeasts or some fungus that are single-celled, it's mitosis and cytokinesis that is responsible for asexual reproduction. But in our other eukaryotes, fungus, plants, animals, it's development, growth, and repair. All right, so that is everything that we need to know about mitosis. Please feel free to use the extra resources on the D2L as well as the study guide. And of course, if you have any questions, please feel free to ask. And thanks for watching. Hope you have a great rest of your day.